Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for UW-Madison's Division of Extension, also known as Cooperative Extension formerly. And together with those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Robert Lemanske. He's a professor in the pediatrics here at the UW-Madison School of Medicine and Public Health. He was born in Milwaukee and went to high school at Nathan Hale High School in West Allis. Then he came to UW-Madison and studied chemistry as an undergrad. He stayed here and got his MD degree. He did a residency here in pediatrics and then a fellowship here in allergy and immunology. And then his mentor told him to go away. Was it that nice or was it a little more gentle?
Close.
He said, go away so you can come back, which is really good. So he went to the National Institutes of Health, specifically the National Institute for Allergy and Infectious Disease. And then he came back in 1983 to be on the faculty, and now he's a professor here in pediatrics. He's also the Associate Dean for Clinical and Translational Research. And he's going to talk to us tonight about something that's pressing to a lot of us who have children or grandchildren. When the sneeze becomes a wheeze, discovering the origins of childhood asthma and how this will lead to a cure. Please join me in welcoming Professor Lemanske to Wednesday Night at the Lab.
Well, it's a real pleasure to be here, and thanks for coming out in the weather. And my previous schedule for coming here was the night of the polar vortex. So I guess in retrospect, I'm glad we called that one off because I'm not sure there'd be anybody in the audience, but I'm happy you're all here. So what I'd like to do in the time that I have is to talk to you about a topic that has been my whole life, and that is childhood asthma. Trying to determine the origins, basically how asthma starts in children, and then going on to, if we think we know maybe what's happening in terms of how it gets started, is there anything we could possibly do to prevent it and actually keep asthma from developing in early life? So to get everyone on the same page, what I'd like to do first is to give you some facts which I think will be very important for you to consider. First, there's close to 27 million persons in the United States that have this condition. There's over 10 million MD office visits. We have close to 500,000 hospitalizations for asthma, so the morbidity is quite high. And unfortunately, we also have mortality. We have over 5,000 deaths per year in the United States. It disproportionately affects children and black children and adults. And importantly, from a pediatrician standpoint, 50% of children with asthma miss 1 or more days of school per year, which leads to about 14 million days of school missed in the United States, which is a huge problem for us. In fact, it's the most chronic cause of school days missed, or chronic disease in terms of school days missed, that children are afflicted with. And in terms of the economic burden, the cost of asthma, at least in the United States, is close to $80 billion and probably more at this point. Now, there are two additional important points that I'd like to add to emphasize. First, asthma has been increasing in prevalence in the past 20 years. And during the question and answer period, I'd be happy to expand on this, but there's a lot of data to suggest that this might be related to the so-called hygiene hypothesis, which really means that we are now living in too clean of an environment, and we're subjecting our children in early life to living in too clean of an environment as well. The second part is asthma usually begins in early life. In fact, when you ask adults, about 50% of adults with asthma will say that they began to wheeze in the first 3 to 4 to 5 years of life. And that's really what I'd like to focus on during my lecture, and that is basically how asthma gets started in this early time period in life. I'm sorry if, there we go. So the title of my talk, When the Sneeze Becomes a Wheeze, will be important for me to try to make these connections for you. So we know from epidemiologic data and from a lot of different observations clinically and also in the laboratory that usually kids, when they're going to go on to develop asthma, begin to sneeze early in life. And if you think about the causes for sneezing, one potentially could be allergies, and the other could be the common cold or respiratory tract infections. So what I'd like to do in the next few minutes is to go over with you some of the interactions that we have discovered between the development of allergic sensitization, or the development of the allergic response, and how this interacts with our host defense against virus infections in the first 4 to 5 years of life, and how this sort of gets the asthma phenotype established to develop in the child by the time they reach school age. So let me answer this question first. How do allergies develop? Well, it begins in a person who is atopic. And an atopic person has the genetic predisposition to the formation of increased levels of the allergic antibody, which is called IgE. So an atopic person really has the genes to become allergic, but that doesn't mean they're necessarily going to become allergic. Then, when the individual is born and begins to interact with their environment in terms of the foods they ingest, the bugs that they're seeing in their environment, exposure to pets, exposure to moles, exposure to house dust mites and plants such as tree pollen or ragweed pollen, they can then turn these genes on. And if they turn these genes on, they develop what's called allergic sensitization. Now, we can test for allergic sensitization in the clinic or in the laboratory. I'm sure many of you may have been evaluated for allergies, and that's why you're here. We can do the skin test, and I think you can see in the bottom left panel somebody who has multiple allergies. And we can also look for this in an in vitro system where we can take somebody's blood, isolate the serum, and then we can actually measure the IgE antibody that could be specific for foods, could be specific for cockroaches, at least in the inner city. This is a major problem. House dust mites in many parts of the world, and pets. And we have some data from our laboratory to suggest that if you're going to get a pet, you should get it before the child is born. And our data would suggest that dogs are more protective against the development of allergic disease, actually protective against the development of certain allergic diseases and asthma, perhaps more so than cats. So when I talk about that particular laboratory result, it makes me really feel good about saying that dogs are indeed man's best friend. So we have the genes, we activate the genes, the person becomes sensitized, and we can demonstrate this by the testing I just went over. But that doesn't mean yet that the individual has allergic disease. The next step is the targeting of this particular response to a organ system. Somebody who has hay fever, it'll be targeted to the nose. Someone who has asthma may be targeted to the lung. And then the thing that we least understand is the same immunologic mechanism can actually kill somebody when they eat a peanut. So that's where we're going right now in terms of research, is trying to understand how this system, this allergic sensitization, this IgE antibody formation, gets targeted to a given organ system. So that's allergies. Let's talk about viral infections, and I'm sure many of you in the audience know this, But what causes the common cold? And we in our group here have done a lot of, work with the human rhinovirus. Elliot Dick here, the late Elliot Dick on campus, Ann Palmenberg, her laboratory, Jim Gurn's laboratory, one of my colleagues, have been looking at human rhinovirus species for decades. And we now know that there are 3 different types of rhinovirus, A, B, and C. We knew about A and B for quite some time, but C was just discovered within the last 10 years. And the important part about C is it binds to a different receptor than does A and B. So if you work for industry and you want to try to come up with some medication that could potentially prevent the common cold, you would like to be able to know the receptors that these different cells, or these different viruses bind to try and prevent the cold from occurring. Now, we know that at least in childhood, respiratory infections are the most common cause of asthma attacks. In children, in fact, up to 80% of asthma attacks or asthma exacerbations are usually related to a viral respiratory tract infection, and most commonly related to the common cold or human rhinovirus. So related to human rhinovirus, what have been some of the important discoveries that have been taking place here at UW? Well, it's hard for me to believe this because when I was a fellow in training, this particular publication was really a very important discovery. And this was by Dr. Miner and colleagues working with Dr. Elliot Dick. And what they did is they looked in the general population here in the Madison area, and they found that the human rhinovirus was involved with asthma attacks in Madison school children. And this was really the first observation that connected the rhinovirus species, if you will, with attacks of asthma in school children. Then Dr. Bussey and I, Dr. Bussey was my mentor, we began to do experiments in adults, infecting them experimentally with rhinovirus, and then challenging them with antigen to see if the infection would alter the response to the allergen challenge. And what we found is, lo and behold, the infection did indeed alter the response to the allergen exposure, such that the individuals that we challenged developed an immediate bronchospastic response, which occurs in minutes, but when we followed them out 4 to 6 hours later, they had another episode of airflow obstruction, which is called the late phase reaction, which is really related to inflammation, which is a very important characteristic of asthma, airway inflammation. So this was the first time that we really could say that an infection in some way altered the response to an allergen exposure. So this then led us to begin to think about, in early life, when and how does asthma begin? And from some work that we performed in an animal model in which we took rats and we infected them with paramyxovirus, which is a rodent pathogen, and we infected them at an early time in life, at about 3 weeks, which is when they're weanlings. And we had 2 strains of rats, one that was an allergic strain, if you will, and one that was a non-allergic strain. And when we infected them both, the allergic strain, 8 to 10 weeks later, went on to get asthma in a rat. And we could tell that by how the airways looked under a microscope. And my colleague, Ron Sortness, could do airway physiology in small rodents, and we could clearly demonstrate that the animals developed airflow obstruction, and they also developed airway hyperresponsiveness, or twitchy tubes, which is a major characteristic feature of human asthma. So we also learned then that, with some additional experiments, that we could alter the immune system from a Th1 response, so to speak, to a Th2 and vice versa. And by doing so, we could take those animals that were allergic and make them less allergic, and we could actually prevent them from getting asthma. So this was a huge observation, and it led us to say these results are so compelling, we have to test it out in the human. So this led us to propose the COAST study, which is an acronym which stands for Childhood Origins of Asthma. And this is a birth cohort that I put together in 1998. And it's a prospective study in a high-risk cohort, meaning one or both of the parents have either asthma or allergic disease. And it's designed to evaluate the interactions among age, patterns of immune dysfunction, and virus and bacterial infections with respect to the subsequent development of asthma and allergic disease. Now, this is how COAST was set up. In 1998, our biostatisticians said that in order to answer the question we wanted to answer, to answer the question that we wanted to ask, was, and this was to look at which kids developed persistent wheezing when they were 3 years of age. And then in the second graph, when we were lucky enough to be refunded, we looked at them at 6 years of age because we could diagnose asthma by age 6, but we could only diagnose persistent wheezing at age 3. So we had 289 kids that were enrolled. Again, this was high risk, at least one parent with allergies and or asthma. We had the kids normally come for what's called well child visits, and the coordinators met the kids in the clinics when they were seen by their primary care docs, and they obtained a nasal lavage specimen. They actually put saline in their noses, sucked it out, and then we took this to the laboratory, and the Wisconsin State Lab of Hygiene was kind enough to do virology on these specimens so that we could see the background carriage rate of viruses in the community of these kids in early life. We also did nasal lavage specimens during symptomatic illnesses. And when I think back about this, it was absolutely remarkable how we conducted this. This was an R01 grant, and I only had $250,000 a year to be able to conduct this study until I turned it into a program project grant 4 years later. But the coordinators were so devoted to this study, they would actually go into the homes or go into the clinics when the kids were being seen for sick visits, and they would perform a nasal lavage to be able to detect what the pathogen was that was causing the problem. In doing so, we were able to record the timing, the severity, and the etiology of respiratory illnesses throughout childhood, which was an extremely new and important set of observations. We also, through blood testing, as I showed you previously, were able to do a longitudinal evaluation of the development of allergic sensitization. When did the kids become allergic, if they were going to become allergic, and what did they become allergic to? And this was very nice for the parents because they were getting all this testing done for free, and we communicated all this information back to the primary care docs, so they were getting all this information at the same time. Then we were able, at age 3, at the end of this R01, at the end of 4 years, we were able to look at the development of persistent wheezing. We published these results in 2005. And then, happy to say, a 90% retention rate by the time the kids were 6. We were able to determine which of these kids went on to develop asthma by the time they entered school. Now, what did we learn in terms of what these kids were wheezing with in terms of the viral pathogens in early life, and those kids who went on to get asthma when they were 6, and those kids who did not? What we're graphing here is the mean number of wheezing illnesses per year in year 1 in black, in year 2 in gray, and year 3 in the open bar. And the various viral pathogens are listed here: novirus, rhinovirus, respiratory syncytial virus. I can't see this. Parainfluenza virus. Thank you. Influenza virus, coronavirus, metapneumovirus, adenovirus, and enteroviruses that at that point were not classified. Now there's two important things about this graph. The first, if you look at the overall respiratory pathogen burden, it's much higher in the kids who went on to get asthma versus those that do not. And if we specifically look at the number of wheezing illnesses related to rhinovirus from year 1 to year 2 to year 3, it's going up each year. In contrast, the exact opposite pattern is being observed in those kids who don't go on to get asthma. Their chances of wheezing with rhinovirus are becoming less and less from year 1 to year 3. And the difference between these two patterns is highly statistically significant. We also looked cross-sectionally at those kids who are allergic at age 3 versus those that were not. And a RAST test is an abbreviation for a radio-allergosorbent test, and we can actually measure antigen-specific antibody, IgE in this case, to various respiratory pathogens. So if a kid was RAS positive, they had allergic sensitization. If they were RAS negative, they did not. We also looked at the interaction between these two events and the development of asthma at age 6 years. And what we find here, this is the comparator group, those kids who never wheezed with rhinovirus in the first 3 years, and those kids who were not allergic at age 3. Those kids who were allergic at age 3 but did not wheeze with rhinovirus, you can see here that this particular development, becoming allergic, does increase your risk of going on to getting asthma. If they wheezed with rhinovirus, but they were not an allergic at age 3, their odds ratio of going on to getting asthma were substantially increased. And if they had the double hit, if you will, they wheezed with rhinovirus, they became allergic by age 3, their chances of going on to getting asthma were huge. So, from what I've showed you so far, what have these findings in COAST taught us? Well, there seems to be, in terms of asthma, both inception and having asthma attacks once you get asthma, that contributing factors are the development of allergic sensitization in terms of antigen-specific IgE antibody formation and the development of virus-induced wheezing illnesses. So an important piece that was unanswered when we were doing this particular study in those first 6 years is which is the horse and which is the cart. Does allergic sensitization precede the development of virus-induced wheezing, or is it the reverse? Does the virus get in the lower airway, strip the airway epithelium, the surface of the airway cells in the lower airway, and in doing so allow antigen to penetrate through the mucosa of the airway, activating immune response cells and allowing the kid to become allergic? And what we found in some very cool modeling that our biostatisticians did is that definitely allergic sensitization precedes the development of virus-induced wheezing illnesses. Now, there's a tremendous interaction between these two, and we've gone on to look at mechanisms in which this occurs. Now, we know when somebody becomes allergic, their host response to virus infection changes. Their ability to produce interferons, which is a major cytokine which dampens the response to viruses and prevents or at least attenuates viral replication, is significantly attenuated. And there are mechanisms for that, and I can draw them for you on the board if you're interested. But this has now been demonstrated not only by our group, but by other groups. And so the development of this sensitization process in some way influences the host response to viral illness. Now the next question we wanted to answer is, can genetic findings be even more informative? We looked at host response, we looked at the environment in terms of allergic sensitization. We now wanted to drill down to look at genetics and whether or not either an isolated genetic defect or some type of gene by environment interaction might influence some of the results that I've shown you. Well, okay. So what we looked at asthma susceptibility genes and their relationship to rhinovirus wheezing illnesses. And for those of you who aren't geneticists, we looked for the genes that code for asthma development in early life. And what we found is they could be linked specifically with RV childhood wheezing illnesses. And the locus, the genetic locus that seemed to be the most important, at least so far, by GWAS analysis, meaning you look at the whole genotype, is a locus at 17q21. So you're probably asking, well, what does 17q21 mean? Well, if you look at human chromosomes, and this is a human chromosome in green, and then you look at a specific locus on the chromosome, in this case, the important locus for us was the 17q21 locus or region, and then you can break apart this locus into single nucleotide polymorphisms or SNPs. And what we did, and we published in the New England Journal of Medicine in 2013, is we looked at asthma prevalence based on genotype at a certain locus within the 17q21 region. So these are kids who, when they get one component, one allele from the mother, one allele from the dad, these are kids who were homozygous CC. These were kids who were heterozygous CT, and these were kids who were homozygous TT. Now, the first one thing I want you to see is if you look at the blue line, these are kids that did not wheeze with rhinovirus in the first 3 years of life. So regardless of what genotype they had at this particular SNP, their chances of going on to get asthma were absolutely no different. In contrast, if they were homozygous, they had one T, or they were homozygous TT, their chances of going on to getting asthma were huge. In fact, kids who were homozygous TT, 90% of these kids who wheezed with rhinovirus in the first 3 years of life went on to get asthma. And the importance of that is that if we want to design studies, therapeutic studies, and you're a parent, and we're going to do some things with your child that you may say, well, how do you know my kid's going to get asthma? Well, if we have things like this that could help us predict risk, it would be very important to that message to relate to families, to allow them to really feel more confident that putting their child in these kinds of studies would be potentially very helpful. So from what I showed you so far, how can our COAST findings combined with other results inform us? Well, this is a slide that I put together for one of our journals. We know that there are genes that are important. I've only had time to talk to you about the 17q21, but there's another locus called CDHR3, which is actually a gene that codes for the receptor for human rhinovirus C. We're very, very intrigued by this, and our laboratories now are in the process of designing experiments to be able to really drill down on this finding. But there are other loci that have been described that may influence either asthma development or asthma severity or asthma exacerbations. Now, in terms of the environment, I went over with you viruses, at least the human rhinovirus. We are now doing some work with bacteria, and in many cases there seems to be, they can act as cofactors to make the child sicker. And this is a topic for a whole, probably, seminar, the microbiome and what influences certain environments, particularly living on a farm, seems to be very protective. If you as the mom are in the barn working with the animals and the child, you take them to nurse them or put them in the barn where you're doing other tasks, it's extremely influential in actually preventing asthma. In fact, some of our colleagues, I'm losing my voice, I'm sorry, some of my colleagues in Australia, where the barns are linked, not Australia, Germany and Austria, where the barns are physically linked to the houses, the rates of these kids going on to getting asthma is about 0. Pretty important. Now they're trying to figure out what's in the microbiome that's good and what's in the microbiome that's bad. And we're trying to figure that out too in some of the work that we're doing. Now I also, there's people can develop allergic sensitization, people can wheeze with different viruses, but that doesn't necessarily make them get asthma. So we also think that there probably are critical developmental windows, either in the development of the lung or the immune system, in which these events have to interact to be able to trigger the development of the asthmatic phenotype. So finally, hearing what I just had to say, I'd like to give you an idea of some of the prevention trials that we're currently involved with here at UW. So the 2 trials we have currently in progress, the one is called the PARC trial. This has been funded by the NIH. We are one of 10 clinical centers. And what we're doing here is we're taking kids who have a pretty, not definite signal, but kids who are at high risk. They may have eczema. They have a parent who has allergies or asthma. And we're actually treating them with an anti-IgE antibody between the ages of 2 and 4. This antibody is commercially available. It's called omalizumab. It's been out for a long time, a good safety record, and has been extremely valuable to us in treating patients with more severe asthma. So the thought here is if we can stop the development of the allergic sensitization process, we can actually prevent the response, the reduced response of the host to viral infection, and ultimately prevent the development of asthma. The second study we're doing, well, we're not there yet with viral vaccines, but there are many centers that are currently thinking about this and doing some preliminary work. But we are also involved here at UW with a trial called Orbex, in which we are feeding children between the ages of 6 and 18 months with a lyophilized extract from bacteria. So these are kill bacteria that we're having the kids ingest to change their microbiome. And there's data to suggest that if you alter the gut microbiome, you can ultimately also alter the microbiome of the lung. So I'll show you a number you can call if you have any children or grandchildren that you think you might like to enroll in these trials. These are very important studies based on the data that I showed you, not only from our laboratories, but from other laboratories in the world. Now, I'm the lucky guy that gets to come to talk to you on Wednesday night. But this is just a small number of people who have been my colleagues and friends through the years, who have helped to contribute to over 85 publications from COAST. We've also presented data from the COAST project in 5 continents, which we're pretty proud of. I'd also like to thank the parents of and the children. The children now are 18 to 19 years of age. I've actually seen kids in the COAST study, and they asked me, when I get married and have babies, can my child be in the COAST study? So they've been taken very good care of by our coordinators. So finally, if you're interested at all in knowing more about what we do, and not only the studies that I just showed you, but we also have ongoing studies in adults. And if you call this number, you'll get the Pediatric Asthma and Allergy Research Program. But if you're an adult and you want to know more about the trials that we're doing in the adult population, these coordinators will clearly get you to the right place. But if you have kids who you think might qualify, we'd love to hear from you so that we can hopefully find a cure for childhood asthma. With that, I'm going to stop, and I'd be happy to answer any questions. Yes, sir.
I'm interested as an asthmatic, as you can tell maybe, as I'm talking. You kind of hinted at longevity. Is there anything associated with longevity in terms of having asthma as a child as you get to be an adult?
Well, asthma has both an age and a sex dependency, which is really interesting. Boys outnumber girls with asthma about 3 to 1 in the first 10 years. During adolescence, girls have new onset asthma. Boys tend to lose it or go into remission. So by the end of adolescence, the prevalence between males and females is about equal. But then in adult life, females tend to outnumber males by a little bit. But more importantly, from a clinical perspective, females tend to have more severe disease. So you're unfortunately one of the folks who didn't go into remission, but and it's very difficult to tell in early life which folks are going to keep it and which folks are going to get it to go away. I've also seen adult patients where they have gone into remission and they're doing great, and then they get this horrible respiratory tract infection and their asthma comes back.
I think that's why I'm in remission, basically. I have asthma, I exercise-induced asthma, that's about it.
Okay. You're lucky. That's good. Well, not lucky you have exercise-induced asthma, but the fact that you don't have anything more than that, and I'm sure you probably have some medications that you can work with that will help you prevent having a problem. And if you don't, please come and see me after that. We have, at the 1984 Olympics, there were 500 U.S. athletes. 90 of them had asthma, and they won 60 medals. So just having it should not prevent you from doing what you want to do. And that we really stress in the clinic with the kids. Because we've had parents who have kept their kids out of gym because they were afraid that they weren't going to be able to breathe. And then when they stay out of gym, then they don't go outside. When they don't go outside, they start gaining weight. And it's a vicious cycle. So you got to make sure that you get on top of it before it gets to be too late. Other questions? Yes?
Is there any relationship between prevalence of childhood asthma in a family and whether or not people who don't develop it as a child may develop it later on as adults?
Yeah, that's a good question. The pattern of development doesn't run true from SIB to SIB. Certainly, it runs in families, but it's a complex gene. It's not like CF where there's one particular SNP that can cause, or one particular genetic loci that can cause problems. It's not autosomal recessive or dominant. It's complex. So it's probably multiple genes. And that's another thing that we're working on in our research is asthma is not a single disease, just like anemia isn't. The phenotype for anemia is having a low hematocrit and hemoglobin, and people are pale and tired. People with asthma wheeze, they cough and they get short of breath. That's the phenotype. But if you look at all the causes of anemia, sickle cell, iron deficiency, G6PD, thalassemia, that's drilling down to a mechanism. That's called an endotype. And there you can really be specific about your therapy in terms of what you want to intervene with. They're all anemias, but the cause of them is different. And so what we're trying to do in asthma is we know the phenotype. Now we're trying to find the endotype. And that's really looking for a mechanism. Tom?
I'm interested that you said it's one phenotype in many diseases in the reading we did in the run-up to this. I never heard that mentioned. And
I'm sorry, Tom, I can't hear you. The lights are too bright. Gotcha.
the lights, that's me. In the reading that I did in the run-up to this, I didn't read anywhere else that asthma is just a phenotype and it's actually many diseases. Is that something that you'd like to get out to more people, that it's not just a disease, it's one set of symptoms for many possible diseases? Because that's news to me.
Right. Probably, you know, when we did, I'm sure many of you know what a phenotype is, but a phenotype is the characteristics, the physical or physiologic characteristics of an individual. Tall, short, blue eyes, brown eyes. And with asthma, we have described certain phenotypes. For example, exercise-induced asthma is a phenotype. Work-related asthma, people who get asthma only when they're in the workplace. That's a different phenotype. Virus-induced asthma is a particular phenotype. Allergen-induced asthma. No, that's an example of that would be college students who never have problems with asthma until they go to their girlfriend or boyfriend's house who has a cat. And they're allergic to the cat, and they're miserable. But the mechanisms for each of those, we really have to drill down and find out what the exact mechanism is to generate that phenotype. Yes?
Yeah, you mentioned of course, at the beginning that you know that the sensitivity to allergens is a cause of viral-induced sensitivity. Is there a mediating something you've defined that mediates this cause and effect?
The question, and I'm sorry, I apologize for not repeating the previous questions, but I was supposed to. The question is, allergic sensitization, does that precede or have something to do with the response to virus? Is that?
Yeah, you mentioned this as a
right.
cause and effect, but I was wondering if there's any mediating variables between it rather than a direct connection between the two, or is there something that's been defined, some things between the two that's been defined that mediates that cause and effect?
We do have a mechanism that's been shown in a number of laboratories. There are cells that circulate in the peripheral blood that are called plasmacytoid dendritic cells, and they have receptors on their surface for IgE antibody, and they're very important for host defense to respiratory pathogens. And if you increase the amount of IgE, it'll bind to the surface of these cells. And as more IgE binds to the surface of the cells, when these important host defense cells see a virus, their ability to become activated and secrete what are called cytokines or interferons is impaired. So the host then is unable to exert a proper, if you will, immune response to that particular infection. And we've demonstrated that with rhinovirus, and our colleague Michelle Gill from Texas has actually demonstrated this with influenza as well. So it may be not just alone with rhinovirus, but it probably could apply to other viral infections as well. And that's where the anti-IgE comes from, because anti-IgE will it'll strip all the IgE off the cells, and they're then excreted in the urine. And so the circulating level of IgE goes way down. And eventually you get to equilibrium where the IgE that's bound to cells, when it can't see any amount in the blood, it pops off those cells and goes into circulation. And then this antibody can find it and get it. And eventually you'll strip all the IgE off the surface of the cells. It's pretty cool. We have a lot of biologics now that we have the possibility of using that could also intervene in some of the other pathways that we think are important. Oh, yes, sir.
Have you looked at children with learning disability and whether any percentage of them have asthma? Because asthma will restrict the amount of oxygen transaction in the lungs. So whether that affects the neurological development,
right.
Whether that contributes to learning disability, is there a correlation? And if so, what is the amount of correlation?
The question was, does a child with asthma have a reduced oxygenation because of their lung problem, and that potentially translates then into other kinds of issues such as learning disabilities and things like that. And the answer to that is that most people with asthma, if you measure their oxygen levels in their blood, they're perfectly fine until they get an attack. And then the asthma attack, they develop airflow obstruction, blockage from smooth muscle constriction in the airway, mucus production, inflammation, swelling of the tubes. But eventually that goes away, and that amount of time that they're spending in a hypoxic state, unless they're really moribund, is not going to cause any problems long term. Yes?
Could you update us on any research that's going on, allergy related, with the tree nuts and the peanuts?
Yeah, we actually have a new faculty member with us in pediatrics. Her name is Anne-Marie Singh, and she was a fellow with us previously, and I was one of her mentors. She's just a great person, very smart young individual. Anyway, her main interest is food allergy and atopic dermatitis. We traditionally, our group has been asthma throughout the decades of our program. So she brings in a new interest, which we think is really going to expand our edits, if you will. And we were just declared a FAIR Center, which stands for Food Allergy Research and Education Network. There's like over 60 in the country. And so this will now give us an opportunity to participate in food allergy trials. Now, those centers that already have this ongoing, they are trying to work out a number of different things. One is, why do certain foods cause people to become allergic? Nuts, peanuts, seafood, and milk, eggs, soybean. And others, never. You never see an issue with these. And that may have to do with how the antigen, the food antigen, looks to the immune system in terms of what we call epitopes, which are parts of the protein that are sticking out and allowing the immune system to say, I don't like this person, and then developing an immune response to that. So we're trying to characterize food antigens to see if we can make them less allergic. And if we can, we could potentially feed these to people and turn the immune system away from an allergic response to a non-allergic response. We're also in people, there was a big publication in the New England Journal of Medicine about 4 years ago. I don't know if you remember this, but the American Academy of Pediatrics recommended for years that we restrict the ingestion of peanuts, milk, or peanuts at least, nuts, high-risk foods from the child's diet for the first maybe 6 months to a year of life. So everybody did that if they were following the American Academy of Pediatrics. And then there was a researcher from the UK who was very interested in peanut allergy. And what he did is he gave a lecture in England, and it was full of docs, and he said, how many of you out there have a patient with peanut allergy? I just want to see how many of you deal with this. Just about everybody's hand went up. Then he went to Israel, and he gave the same lecture, and he asked the same question. 2 hands went up in this whole audience. And all of a sudden he thought to himself, my gosh, something's different about what they're doing in Israel compared to what we're doing in the UK. And to make a long story short, when he talked to the people about their culture in Israel, it's a custom for them to give their kids at a very early, probably in the first week, something called a bamba, which is a pacifier that contains peanut protein. So the early introduction of this peanut protein allowed these kids to develop tolerance. And so now the recommendations of the American Academy of Pediatrics are totally different than they were before. So with luck, in the next 10, 20 years, we're going to see the prevalence and incidence of at least maybe peanut allergy going down. There's also desensitization trials in people who already have developed peanut anaphylaxis to give small amounts of peanut protein graded with people under close observation in the clinic to give them increasing doses of the peanut so that they're able to tolerate maybe 2 or 3 peanuts. Now that doesn't say they can eat a whole bag, but if they accidentally ingest a peanut, they're not going to die. And then the third or fourth, how many ever I named for you, is a patch, where they're actually putting patches on the skin with peanut protein, other food protein, to get the thing to be absorbed and to desensitize the patient, if you will, that are allergic to certain foods. All of this is research that's ongoing right now. And there's a tremendous amount of interest in food allergy right now. So I think in the next 5 to 10 years, we should have some really significant breakthroughs, which is good. Tom?
Is there any possibility of doing gene editing of people who have allergies such as peanuts, something that's life-threatening?
Not thus far.
What would it take?
Well, you'd have to come up with some way of being able to alter those genes that are becoming activated and turned on to develop IgE antibody to that protein. 20 years from now, we might be there, but we're not there yet.
But there's nothing that leaves to your mind going, this is the
No.
crossroad that we could look to.
No. But this early introduction of foods, I think, could make a big difference.
Bypass all the others.
So if you want to know where you can get Bombas from, I think we actually ordered some in our clinic because we were doing some studies with them, but I think you can get them in the U.S. I just don't remember where. But.
Isn't there a song like that in the late 50s?
I'm sorry, what? Okay. Okay, back there.
I wonder what dependent variables you're collecting, like the gut biome and the DNA, and which ones you think that you're missing and you may be collecting in the future.
Well, that's a great question. We actually are now part of a multi-site consortium of people in the U.S. that have had birth cohorts. And so Coast is one of them, and we have another cohort in Wisconsin called WISC at the Marshfield Clinic. They're looking at farm environments and the protection from them. But anyway, there's 12 different birth cohorts in the U.S. that have now come together. We are funded by the NIH, and we're in the process of harmonizing our data. And we will be one of the sites here in Madison. We're going to be recruiting new children, newborns, to be part of a new cohort of kids moving forward. And we'll be able to collect specimens that we couldn't or we didn't think of collecting previously. And stool is definitely going to be one of them. And there are sites that already have collected stool that have those biobanked, and we're going to go back and look at them. And there's other sites, too, that are looking at this already in terms of the microbiome. So it's ongoing work. Yes?
Could you talk more about the hygiene hypothesis and its relation to asthma and allergies?
Sure. There was just, for those of you who like to get more into this, there was just a review article on this in the New York Times, probably within the last week. And this was proposed a number of, not the hygiene hypothesis per se, which was proposed in 1989 by a man named David Strachan, and I'll come back to that in a minute. But in the late 1800s and early 1900s, people were observing that hay fever was much more commonly seen in people who were wealthy compared to those who were not. And so people started thinking that this is a disease that's present in people who live cleaner lives, who have more rich things. They didn't know exactly what was going on, but there seemed to be a difference between a more poor rural lifestyle and being in a rich environment. So then David Strachan looked at a whole bunch, I don't know, thousands of patients that he had in a data set. And what he found is that the chances of a person developing allergic rhinitis or hay fever in a family was much less if there were more siblings in the family. So the more kids you had, the less chance you had of becoming allergic, particularly the kids who are at the bottom, the kids who were born last. So that was the original hygiene hypothesis. He related it to the kids who were the oldest were bringing home infections. They were infecting all these kids, turning on their immune systems to be able to fight infection, and therefore preventing them from becoming allergic. Now, the hygiene hypothesis has many different corollaries in terms of the diet, other things we're seeing in our environment, but the bottom line is that the fact that allergic disease and asthma has been increasing over the last few decades probably has nothing to do with genetics, because it couldn't possibly, those changes could not have occurred in some kind of a genetic mutation in that period of time. So people have really turned to the environment as being the culprit, and or one of the culprits, and that's where a lot of the interest is going at this point. But look at the New York Times and see what you think. Yes?
What is the role of all these chemicals in our environment in terms of increasing sensitization to things that we call allergies?
Yeah, that's not stuff that we have looked at in COAST, but I know some of my colleagues in this new consortium that we have looked at that. The question was, what about chemicals in the environment and their effects on some of the things that we've been talking about? Pollution clearly has an effect, and it probably is important, at least in some environmental situations, of turning on the IgE response. And then there are other chemicals that we can ingest, and I can't remember exactly what those chemicals are off the top of my head, but they also may be involved in this as well. So, yep, that's another area, the diet. Vitamin D has received a lot of attention, and there's data on both sides of the fence as to whether or not giving more vitamin D is helpful or not. We looked at it in COAST, and we couldn't see any effect. But other groups, a big group from Harvard, has kind of shown some effect. So, wish we would have seen more effect because we're big vitamin D people here at Wisconsin, right? Yeah. Yes?
I saw in one of your slides a child using an inhaler.
Uh-huh.
Now, do you interact with the pharmaceutical companies from your research? What's the most effective delivery system for the medicine to make it long-lasting or to make it a cure ultimately?
The question was, when patients, I'll try to synthesize this, when patients are using their inhalers for asthma, what is the proper way of being able to administer it? And the answer to that question is that there are two things that are probably the most important: a slow inspiration, and then once you get the medicine in, to hold your breath for 5 to 10 seconds. So what you don't want to do is take the inhaler and go like that, because all the particles will be in your tonsils and none will be in your lungs.
That was not my question.
Oh, I'm sorry. I thought it was.
My question was whether with your research, are you interacting with the pharmaceutical companies which develop these inhalers and medicines, whether so that they can produce more effective medicines
right.
or therapies that can provide a cure ultimately, or is there anything else going on?
The question was, is our research group involved with studies that are being generated through industry with inhalers and other medications that could potentially affect asthma or help it or maybe cure it? And the answer to that is yes. We have a huge clinical research program that we have where we're doing phase 3, phase 4 trials. And there are many new medications, not many, but some new medications. The biggest new advance have been the biologics, the ones that are monoclonal antibodies that will bind to certain aspects of the immune system and prevent it from turning on. And that's actually a very exciting area. The problem with biologics is they're so expensive. 1 year of treatment with a biologic could be between $20,000 and $50,000. And so before you start people on that therapy, you want to make sure that you're selecting the right patient for the right medication. Not only the right medicine, the right patient, but the right time. Yep?
I have a question. What do you think of Primatene Mist coming back on the market?
The question is, what do I think of Primatene Mist coming back on the market? Oh, man. When I was fortunate enough to be president of our international organization in 2015, and we were dealing with this with the FDA. And, the problem with epinephrine is it's a quick fix. It's a very potent vasoconstrictor, so it takes away the edema, and it will cause the smooth muscles in your airway to relax. But it only lasts like 15 or 20 minutes. And there is lots of data that as you keep using these things, you basically desensitize the beta receptors, which is the receptor that the drug acts on, so that you need more and more and more and more. And what that should be a sign of is that you're in trouble. And you need to be seen by a doctor, because if you don't, you're going to die. And then the proper instructions on how to use an inhaler. When somebody buys something over the counter, the pharmacist isn't giving them instructions on how to use it. So there's lots of downside to it. On the other end, there are people who can't afford to see a doctor, they can't afford to get prescription medicines, and they can't breathe. So it's a hard question. There's not a black and white answer.
I just have to comment. For me, it's been a wonderful thing, because I have the exercise-induced asthma, so I'll use it. I'll actually spray it in front of me, and I just tried it when it came back on the market. I'll spray it and then inhale, because I only want a small amount. And that's all I need.
Okay.
Yeah. No, but there's no other hump after that.
Well, just keep in mind if you're running a long race that's only going to last for like 20 to maybe 30 minutes, and that's it.
It'll last longer.
Well, there's also athletes that can run through their asthma because you get what's called a refractory period, where you get a small amount of bronchospasm to develop and then you relax because of release of prostaglandins. And people, very competitive athletes, we can use that. That's one of my areas of interest is exercise-induced asthma. That's a trick that you can use when you're coaching athletes to use the refractory period to their advantage. I don't know if that's happening to you, but maybe it is.
That's interesting. That might be interesting.
Yes?
Could you comment on the use of corticosteroids as far as either the inhaler or pills?
Sure.
Yeah.
The question was, could I comment on corticosteroids both in the form of an inhaler or in the form of a pill? Corticosteroids, inhaled corticosteroids, were just coming out when I was beginning my fellowship training, and they totally revolutionized the treatment of asthma in terms of reducing morbidity. Now, in children at least, there are some side effects if you use too high of a dose for too long. You get growth retardation. But we've done enough studies on this to be able to know where the sweet spot is in terms of how much to give to keep the kid under control versus not giving too much to get side effects. Now, oral steroids are very helpful to us for treating asthma exacerbations when people have asthma attacks. They can be very helpful for that. But again, you don't want to use too much. It's like everything else in life. Moderation. Any other questions? Yep.
Have there been any studies done on why certain races are more affected by asthma than others?
we, meaning the clinical research consortium that I have been involved with the NIH, have done studies, and we still don't have an answer to that question. We just completed a study in which we looked at a totally African American population based on their reports in terms of saying that they were black, but we also did genetics on them, so we could look at genetic ancestry to see what part of the world they came from, to see if people with a certain genetic background would respond differently to a medicine with people who were from a different background. And we really couldn't demonstrate any differences in that study. And that has just been submitted for publication and it's under review. In addition to the black population, we also know that Latinos also have more of a risk, particularly people who come from Puerto Rico. Much more so than Mexico or Central America. But the Puerto Rican population really has bad asthma. And when they move to New York, there are just pockets of people with very severe asthma. Most of them are Puerto Rican. And we don't know the reason for that either, but they definitely, that's a risk for them. All right.
Thank you very much.
Thank you.